Welcome back to another explainer. Today, we're unraveling a massive biological secret using something you probably have sitting right on your kitchen counter. Yep, a potato. We're going to investigate the hidden microscopic behavior of cells and discover exactly how they interact with their environment. Okay, let's dive right into this. Have you ever wondered what actually happens when a living cell meets a giant pile of salt? Or, a heap of sugar? It sounds like a simple question, but the answer literally unlocks the mechanics of how life sustains itself at the most fundamental level. And to figure this out, we aren't just going to read about it. We're getting hands-on. Section 1, the potato experiment. So we're stepping into the shoes of a scientist today to recreate a classic biological setup. In our source material, this is known as Activity 2.2. By setting up this deceptively simple potato experiment, we're going to see firsthand how plant cells react to drastically different environments. Here's exactly how it works. First, grab a standard potato and cut it into two roughly equal pieces. You'll want to carefully measure and record their initial weights so we have a solid baseline. Then comes the critical step. We put one potato piece into beaker A, which is just plain water. The other piece goes into beaker B, which contains a highly concentrated 20% salt or sugar solution. After that, we just leave them undisturbed for about an hour and then measure their final weights. The environment is literally the only difference here, plain water versus a heavily salted or sugared solution. Section 2, swell or shrink. And this brilliantly illustrates the invisible forces at play. Looking at the results, the difference is just incredibly striking. Notice how in beaker A, the potato piece sitting in the plain water actually swells up. It gets noticeably heavier and firmer. But in beaker B, the piece submerged in that 20% salt or sugar solution completely shrinks. It loses weight and gets all limp and rubbery. We started with two identical pieces of potato, but got completely opposite reactions based entirely on the water they were sitting in. Section 3, the science of osmosis. To understand why our potatoes behaved so differently, we have to zoom in. What we're witnessing on a macro scale is actually the microscopic science of osmosis. This is the movement of water across a biological boundary, and it's driven by what we call solute concentration. Basically, solute concentration is just the amount of stuff, like salt or sugar, dissolved in the water. Let's break down the three possible scenarios for a cell in any liquid. First up is an isotonic solution. In an isotonic state, the solute concentration is exactly the same in the extracellular medium outside the cell as it is in the intracellular medium inside the cell. Because there's this perfect, peaceful balance, water moves in and out at the exact same rate, so the cell size, it stays exactly the same. But what about beaker A? Well, that environment was a hypotonic solution. Here, the solute concentration outside the cell, in that plain water, is much lower than the concentration inside the potato cells. Because the inside of the potato cells had way more solutes, water naturally rushed in to try and balance things out. That massive inward rush of water is exactly why the potato piece in plain water swelled up. So, the absolutely crucial point here is what happened in beaker B. That 20% salt or sugar solution created a hypertonic environment. The solute concentration outside the cell was massively higher than on the inside. To achieve balance, the water was literally drawn out of the potato cells and into the salty beaker. As the cells rapidly lost their internal water, the entire potato piece shrank and lost weight. Part 4. The ultimate gatekeeper. All this rushing water brings up a fascinating question, doesn't it? How does the cell keep its internal contents securely inside while selectively letting water pass through? The answer is the cell membrane, also known as the plasma membrane. It's selectively permeable, meaning it specifically chooses what gets to pass through and what gets blocked. And get ready because this is wild. 7 to 10. That is the thickness of this mighty protective boundary in nanometers. Just 7 to 10 nanometers. To put that into perspective, a single nanometer is 1/1,000,000th of a millimeter. This boundary defines the very individuality of the cell, protecting its contents and managing its complex relationship with the whole outside world, yet it is almost unfathomably thin. Section 5, the fluid mosaic model. Now, how can something so impossibly thin be so smart? Scientists explain the structure and function of this boundary using a widely accepted framework known as the fluid mosaic model. Let's break down this incredibly complex barrier. First, you have the lipid bilayer. These are two layers of special fat molecules where the water-attracting heads face outwards toward the environment and the inside of the cell, while the water-repelling tails hide safely on the inside of the layer. Next is fluidity. You see, this isn't a rigid brick wall. These fat molecules can actually move sideways, they can flip, and they can rotate. And embedded right within this shifting sea of fats are proteins acting as literal gatekeepers, helping specific substances pass through that can't get past the fats alone. Finally, because these various proteins and fats are arranged together in a beautiful, ever-shifting pattern, we call it a mosaic. It's a fluid, moving mosaic of fats and proteins, literally keeping the cell alive. We've seen exactly how this incredibly thin cell membrane dictates water movement, causing our experimental potato to either swell or shrink, depending entirely on its environment. But remember, plant cells, like those in our potato, have an extra rigid outer layer called the cell wall that sits outside the cell membrane. This rigid wall is what keeps the plant upright, even when its inner cell membrane shrinks away from it during intense water loss in a hypertonic solution. So I want to leave you with this highly provocative question directly from our source materials Pause and Ponder section. What consequences would you predict for a plant cell if its rigid cell wall were to suddenly become as fluid and flexible as the 7-nanometer cell membrane we just explored? Think about that potato. Think about the trees outside your window and how the physical architecture of biology is perfectly balanced to sustain life. Thanks for joining me on this explainer, and keep exploring.